So 20 minutes late, but we just have discussion and reception at the time, so I think you'll all be happy to wait for that. We start the next session with Nicholas Brown. He's going to tell us about thermodynamics of subcellular compartmentation and its evolution implications.
Okay. So thanks to Axel, Anthony, for asking me to come. I'm going to talk about compartments. I should say that we weren't really directly thinking of astrobiology for this, because I'm working with a biotechnology group, but it turns out it may have some relevance to astrobiology in the sense of the origins of cellular life. So compartments, of course, will be one of the main themes in the origin of life, and the compartments underpin the distinction between the major cell lineages, prokaryotes on the one hand and eukaryotes. And traditionally, debate is always focused on membranes in the context of compartments. So what I guess my message is going to try and be is that membranes aren't the whole story of compartments, because you also have this thermodynamic mechanism for compartment formation, which I think to the physicist is going to be rather banal and obvious, but I think it's something that isn't really hasn't really featured to the extent it probably should in the molecular biology of cell biology. So that's my talk organization. I'll talk about TC and prokaryotes. Any kind of compartmentation in prokaryotes is quite significant, because the conventional wisdom is that prokaryotes don't really have compartments, and that's what underlies the distinction to eukaryotes. So this is a little outside the mainstream, but on the other hand, there is a sort of consensus forming that there are compartments in prokaryotes. So I'm not really that far away. Physiological control of the thermodynamics is going to be important. And then finally, I want to get on to the issue of the evolutionary relations between prokaryotes and eukaryotes that are suggested by this mechanism. And then please butt in. It's quite short, so I don't think it's a time issue if you have a question or something to go on. Plus, I'd probably say something stupid and find it out. So I think the cultural backdrop, which is a good starting point, is cooperativity, because cooperativity is something that's quite familiar in protein folding. Basically, it's the idea that protein folding has a thermodynamic component affecting the dynamics of folding, and that's really one of the major reasons why proteins can fold at all, is that the coarse thermodynamics has somehow been tuned by evolution. So various people have started applying that same paradigm of cooperativity to the nucleoid, which is obviously a much larger molecular entity. And the dynamics of the nucleoid can also be understood to some extent as a thermodynamic process. The archetype of nucleoid dynamics is probably transcription, I guess. This is a cartoon of transcription. Out comes a loop, in goes your RNA polymerase, does its thing, and out comes messenger RNA. So how do those movements occur? Well, in a eukaryote, you have histone-like molecules running around, mediating that process in a very mechanistic way. But in bacteria, by contrast, that seems to be a much more coarse physical process to do with the compaction and collapsing of the nucleoid, driven by this what people are calling crowding, which is basically a concentration effect of the surrounding cytoplasm. I guess crowding is sort of familiar in biochemistry, because people have realized that in vitro experiments on cellular molecules don't always work unless you have a specific agent in the in vitro solution that's somehow simulating the much denser environment of real cells. So crowding has sort of picked up momentum in biochemistry generally, and here it's important because it's a compaction effect, controlling the nucleoid cooperativity. One of the main characters who's doing this, well, this is just a cartoon of crowding. There you go. It's just very crudely showing that the express protein complement is the main thermodynamic force forcing the nucleoid to compact. So I think from a physical chemistry perspective, you expect that if you increase the concentration of any kind of liquid system, you expect some sort of phase transition to occur. That's the basic physical chemistry of the liquid-gas transition, the van der Waals theory. So it comes as no surprise that the DNA compaction, in that sense, lends itself to interpretation as a phase-separated part of the cytoplasm. And beyond the DNA, you can extend that concept to include all the other molecules associated with it, the transcriptional apparatus, most obviously, as part of a single phase-separating system that somehow extricates itself from the rest of the cell so that it's able to carry out its biochemical business. So the guy who's looked at this experimentally is a guy at NIH called Kim Zimmerman, and he comes from this crowding type community, and he has a technique to extract nucleoids from cells and denature them in vitro and look at what's inside the nucleoid by separating them on a gel. And not surprisingly, he confirms this idea that phase separated with the nucleoids, with the DNA, are not just the RNA polymerases, but a host of other molecules thought to be associated with transcription. So this is the basic ansatz that bacterial physical chemists can now work with, that they're dealing with a real compartmentation effect that doesn't require remembering. It's a spontaneous thermodynamic effect.
But it's not the same material, right, which is on the two ends, on the two sides of the phase transition. It's DNA.
Not the same material.
I mean, the one is DNA, and the rest of the cell is not DNA.
But I think a phase transition, well, I'll come to the idea of the relationship of phase transition, of a miscibility gap. But I think it's fair enough to say that a phase transition isn't restricted to a single component system as a paradigm. You can have a phase transition in a multi-component system. But I guess you have in mind the sort of single component gas.
Yes, yes.
Yeah.
Okay.
Okay, so that's just that's pulling that together. It now looks as if in prokaryotes, translation and transcription aren't decoupled in the classical sense, as it's often been assumed. They're not decoupled by membrane as such, but they are spatially localized within the cell. And then there is a very nice experiment by Lewis et al., which shows this using fluorescently tagged proteins. This is two cells during the exponential growth phase, showing the ribosomes just beginning to polarize in the cells. And that can be juxtaposed against other molecular tagged species. So they've also tagged the polymerases that are involved with transcription. So here you have the idea that they have is they want to demonstrate the different cellular localization of transcription on the one hand and translation here. And they see that very nicely by superposing those in a single overlay. So you have really concrete evidence experimentally for this compartmentation. And here they've got a similar experiment on a larger length scale. This is a filamentous picture. You have the cell walls, the ribosomes, then you have the nucleoid, or these are the chromosomes. And juxtaposing those two, you see that the ribosomes can't occupy the same space as the nucleoid, so they're excluded. While the RNA polymerases, on the hand, more or less correspond to the nucleoid. Right, theoretically, this is what Axel was pointing out. You bring this into line with the molecular statistics of phase separation, and the unifying concept is a miscibility gap, which basically is the single phase region of coexistence extended to a multi-component solution. This is an illustration. Here's my thermodynamic phase space. This is just a projection, actually, of the larger thermodynamic phase space. There are other parameters that are relevant, like temperature. But crowding is important. So here's the classic sort of concentration effect, that you get a phase transition if you increase the concentration of a gas, you get the liquid onset. This comes out of a calculation that I was involved in. It's not obvious what the effect of protein size will be. In fact, it turns out smaller proteins turn out to be more unstable, and in fact, the phase separate faster than larger ones. Anyway, so this is the issue that the prokaryote has to deal with. That it's somewhere in its phase space is this gap, and if it crosses into the gap, either by, in this projection, an increase in crowding, or some change in expression, changing the mean protein size, then you're gonna see these demixing effects. So, in this example of nucleoid cooperativity, it's a desirable effect, and it's exploited for transcriptional function. But more generally, there's nothing to say that demixing effects could occur any time, anywhere in the cytoplasm, because this gap is hanging around in the phase space. And presumably that's a big problem for physiology. It's as if, at any stage the whole cell can start boiling and then crazy things will happen. So some sort of mechanism needs to be in place to deal with this. So one mechanism which I think in biotechnology is sort of implicit is that you can sort of drug the cytoplasm with clever molecules that are going to have some effect on the overall thermodynamic stability. And here the archetype is osmolytes. You have in many prokaryotes, you have a system of osmoregulation based on compatible osmolytes, which are evolutionarily selected, very small molecules that have quite odd physical chemical properties. So a trick that is implicit in, say, protein expression is to put these compatible osmolytes into the growth medium. The cell, as it starts to overcrowd the cell with this recombinant protein, then uses these osmolytes to try and suppress the miscibility gap, and that can also be shown theoretically. So that is one strategy to deal with the miscibility gap, is to use specific molecules. Strategy B, which is a sort of cold adaptation strategy, is to make sure there's nowhere that a new phase can nucleate. So here's a sort of schematic nucleation site. And then I guess it's obvious to any physicist that you get this what's called heterogeneous nucleation. The geometry plus the specific chemical properties of the surface can induce the nucleation to start, where otherwise it would be suppressed by the surface tension of the growing compartment. So here, this is just showing that without the site, you have this option to supercool to some extent. It's quite hard to model theoretically, obviously, because with a cell you have almost no information about the membrane in particular, which obviously might also be a good nucleation site. So there's a third strategy, if all else fails, which here you're inside the miscibility gap, a compartment is formed, and this presents a rationale for an intracellular membrane, because a membrane gives you immediately many options to control the otherwise volatile compartmentation. You now have the option, for instance, you can let certain molecules through and keep others out. So that's the sort of damage limitation strategy. And that seems to agree with a specific bacterial cell plan that is seen in Phantomycetes, which is they're quite a large domain of bacteria, but they're not typical in that no other bacterial species have these extra membranes. There's the nucleoid. This new compartment is called in Phantomycetes the pyrellulosome, and this is called I think it's called the peripheral plasma. So why is it these Phantomycetes only that seem to show this membrane buffering effect? Well, I think a plausible idea is that it has to do with the very large genome, because they do have the largest genome so far sequenced in bacteria. And that immediately suggests that the genome itself is possibly a nucleation site for a compartment. So this is just a cartoon about nucleation. Any kind of thermodynamic phase separation is associated with what's called a nucleation barrier. In the absence of a nucleation site, the new phase, to appear at all, has to get over, although it's stable globally, it has to still get over a barrier before it can grow and become a macroscopic feature. So if we now treat the nucleoid itself as a nucleation site, a small nucleoid is not sufficient necessarily until it gets over the peak, this critical diameter. So here's a cartoon showing it going over the peak. It's now at the critical threshold, and this is the point at which the compartment becomes thermodynamically real. There it comes, and it grows as it heads for the stable trough on the other side. So the implications for karyote evolution, which I guess is the reason why this is relevant to astrobiology, already they're quite they're quite talked about in evolutionary circles, because these are the planktomycetes membranes. This is a eukaryote nuclear envelope membrane, and there seem to be structural similarities that are quite hard to refute. This is not all planktomycetes have a double membrane to their nuclear compartment, but this is called gemata. It does have a double membrane, which is mimicking the nuclear envelope quite accurately. Not only that, it also has this sort of folded over pore structure. The only feature that confuses people is that there's no homology at all. But I think that's rather moot, because I think the phylogenetic paradigm is rather overrepresented in this issue of cell origins, because it's been so successful in the context of the mitochondrion, for instance. So I don't think it's actually that significant that there's no homology between planktomycetes and the nuclear.
Sorry, I didn't quite follow. What was there was no homology.
Your homology, what do you mean by that?
Oh, sorry. Homology is, well, it's more or less what molecular biologists always do to demonstrate mechanism. I guess they look at the protein sequence in a known context, and they extrapolate that context to another cell purely on the basis of the same information content in the protein.
So it's a one parameter sequence dependent on one parameter?
Well, let's say planktomycetes was the ancestor of a nuclear envelope. Then you would say, well, then it should have exactly the same proteins in its nuclear envelope as the eukaryote. And then, because you have access to that sequence data, you can test that hypothesis. What in this case is the case is that there is actually they're not the same proteins at all.
So, okay, there would be a homology if there was a link.
Yeah. But I think that's actually not a necessary requirement logically for correlation between the mechanisms. I think what planktomycetes seems to demonstrate is that the principle of a large nucleoid seeding the compartment can in principle work for any bacteria that suddenly evolves a large genome.
But in biology you also have the term of analogy. This is more an analogy. The problem is about the same. They found just to make a compartment, and they solve it in the same way. They just use different kinds of materials to solve it.
Yeah, if you like. Yeah.
So it's this is also quite common in biology as well. That things look the same but without being of the same origin.
I think a cytologist wouldn't argue.
Yeah.
But I think because comparative genomics has become so important, those guys do argue, because that's their tool. They have no access to other tools.
So, am I understanding you right in you're saying that it's not a genetic trait, but essentially a common way of solving a problem?
Yeah.
Which can happen regardless of genetic inheritance.
Yeah, I guess it's a sort of physical mechanism. You're looking for a universal mechanistic view, rather than this sort of biological attention to specificity, to the specific details of sequence. So that's, I haven't really got the evolutionary background to follow the implications for the existing state of debate, which is, I know, a good reason to be here. But some obvious conclusions that occur to me are that I think this mechanism seems to me to make redundant one particular aspect of the debate about origins, which is the idea of a mitochondrial seed being required before the evolution of the nucleus. And there are various reasons why people think a seed is required, usually involving the need to decouple translation from transcription, which, you know, this argument about TC, about the use of crowding to effectively compartmentalize those processes seems to make redundant, in my opinion. I think it seems really quite clear now that there is some form of very crude decoupling already in prokaryotes. It doesn't involve membranes, but there is. Maybe I should have made clear that the nucleus in eukaryotes is where transcription occurs, whereas translation occurs outside the nucleus. So that's one of the main themes of any attempt to rationalize why the nucleus has come into being in eukaryotes. So without going into the details of the theory of the mitochondrial seed, that is one aspect of the conclusions. If you have a physical mechanism that doesn't require a seed, then the all-important last universal common ancestor doesn't necessarily have to have a mitochondria. The nucleus could have been there previously. So the selective rationale here disappears. You don't really need a reason to select the nucleus as such, which is quite important because the nucleus obviously has some selective context now. It's subsequently evolved certain biochemical features that obviously have some selective advantage a posteriori, but it's quite important in any evolutionary theory that there not be a positive selective reason, because that sort of begs the question of how the organism knew it was heading for an advantage. So this is sort of a quasi-neutral mechanism. The nucleus simply appears as the genome for other reasons, gets larger. And then that ties in with a sort of much older, larger debate about which way evolution is, the direction of evolution in the context of eukaryotes. Did prokaryotes evolve from eukaryotes, or vice versa? It's quite non-committal in that respect. Evolution could go either way. So that's sort of the tip of the iceberg. I guess that's my take-home message, is I think the nucleus, against the backdrop of this mechanism, could just be a hitchhiker. Okay, so any questions?
Okay, thank you.
Thanks.
And a lot of questions. So I could start by asking about the last thing here. So are you implying that if you have a eukaryote that actually gets streamlined down to beyond 7 megabases or so, it would lose its nucleus, nuclear membrane?
It wouldn't necessarily lose nuclear membrane, but it could do consistently with this theory. I think the theory doesn't, it's sort of very crude, obviously. It's looking at very crude physical, chemical thermodynamics. So it's obviously, it's limited to how complex it can be. Yeah. It's just, it's basically liquid state theory, so I think, you can't obviously say anything about the mechanism of losing a membrane once it's there.
But that's what you say.
I say if you take away the membrane, then and shrink the nucleoid, then you would lose that component.
Okay. Anthony?
So I thought it was fantastic, very interesting from my perspective. Regarding the genome size, you said once you get to maybe the genome size of lactomycetes is what pushes this. But then at the same time, you said that there were, you were describing up until that point a number of other parameters, and presumably there must be a combination of those that can drive this, because I mean there are bacteria with genome sizes of that order that are, for example, among Rhizobium, that don't have compartmentation. So I assume that you can. Is it possible to try and model that in some way? Take different parameters. You know, the genome size will obviously have one effect because it affects the size of the nucleoid, but then you have size of proteins in this graph you showed, and then presumably the concentration of those proteins would also have an effect.
Yeah. No, that's absolutely right. Yeah, you have quite a large parameter space. I think genome size caught my eye because it really is, apart from maybe actinobacteria, I think. I took a brief look at all the bacteria.
Streptomyces, I think, are up to around
7.
Yeah,
Can we
up
take-
to
I think
9.
it's
9
eight,
even.
eight among the Rhizobia, these sort of bacteria.
Pseudomonas as well.
Yeah. So if you-
Pseudomonas as well.
Oh yeah.
So it's definitely up there, but as you say, it's not the only one. And I guess there are other factors. I mentioned the role of osmolyte systems. I wasn't able to check whether, see, not all prokaryotes have compatible osmolyte systems. That was my other candidate mechanism for controlling this. Yeah. I would imagine that if you have that osmotic way to control miscibility, then the genome size becomes more. So I would quite like to check the Planktomycetes genomes to see if they have these osmoregulatory systems. It's not something I could find from the published genome sequence, but I guess in principle I could do it by blast search at some stage. That would probably be quite an important test. But there is a larger parameter. There is some sort of ad hoc space to fool around with this. It doesn't have to be genome size.
Well, I suppose that's two things really. One is, I mean, that's interesting in that, as you mentioned, these Planktomycetes, they have quite different internal membrane structures, which just confuses the crap out of us biologists, because some of them have a single membrane, some have a double. So there's more diversity in membrane structures in that one group of bacteria than there is in the rest of the bacteria put together. So that's confusing. So this makes a lot of sense from that point of view. If it's an event that can happen frequently given certain conditions. And I suppose the interesting thing for the other common mechanism from the point of view of astrobiology, the interesting thing is that would then be something that you could argue is not contingent on certain earlier events in evolution that can happen over and over again, and therefore it may be a feature that we may expect to see in other unrelated life to the membrane, or maybe not even over the membrane.
Yeah, the origins debate does seem to hinge excessively on one-off events.
Yeah, well, I think that's the problem of not being able to approach that material in any other way.
Yeah. Well, I think
it focuses on the traditional biology. Those other areas of outside ASCO.
Yeah.
Excellent.
So nucleation barrier means that is nucleation only above a certain size. Is that what you're saying? And how big would that size be?
Well, I actually can calculate that in principle, because I do have a physical model of all the components.
So how big is that, and what does it depend on?
I haven't calculated that yet, but I think I can do that in principle. That would be interesting. Yeah. But that's definitely something that's not out of the question.
Mm-hmm.
It looked like your cells were pretty empty in the animations that you had done, but you take into account that they are crowded, sort of, that everything is fully crowded.
Well, that was a schematic,
right?
Of course.
Yeah.
Yeah. Crowding is the main parameter that's driving everything. So it's not at all empty. I guess it's empty in the schematic sense, so it's assumed to be very uniform, because that's the way you approach things statistically. You have to make that simplifying assumption.
I also wondered about where you reviewed other papers about the different spacing of transcription and translation in bacteria also. Have those people made sure that the ribosomes are actively translating ribosomes? Because there's a lot of of ribosomes. Half of the cell is ribosomes.
Yeah, I guess their concern was to show that the ribosomes are not entering the nucleoid at all. If there is some coupling, it's occurring at the interface of the nucleoid surface.
Because when you study transcription and translation as a molecular process, you can easily show that there is coupling. Actually, there is even coupling between the different compartments. You can see the interaction between them.
Yeah, but I think, mechanistically there needs to be some there needs to be some sort of arrangement, doesn't there, where things are brought into line with each other. And I think many of the arguments about why the envelope emerged are that you really need to separate them physically, because otherwise, in the wrong context, translation can run ahead of transcription and vice versa. That's causing detrimental effects.
Yeah, or translation can run ahead of post-transcriptional events.
Yeah. Yeah. So that's I guess you've read this Kuhn type approach to things.
How do physically? How do ribosomes know where to go? Is there any? Do they recognize each other? When you see, it was a nice picture showing that you have ribosomes in one side. The nucleus is relatively easy because it's already one big molecule.
Yeah.
So it just has to shrink.
I guess you never assume that ribosomes know where they're going. They just diffuse around in a sort of random way.
But then they suddenly form this crowding pattern.
Crowding is a sort of random statistical effect.
But
the only crowding effect.
But randomly things would dissipate. They wouldn't make it, or they would equally distribute around the space that they have. Somehow this space has to be.
Well, I think that's probably the conceptual thing that's quite hard to come away from.
If it's diffusion driven, then it would go.
Well, that's the hard thing to explain about phase separation.
Okay.
Because you don't need to physically put a membrane between the new forming phase to make it separate. The molecules just decide they don't like other molecules. They want to be together with their own species, archetypically. So they spontaneously start to localize in a clump.
You understand it, if you have lipophilic and hydrophilic things, then
that's an example, yes.
They like the membrane. They do it because that's the way they have to do it.
Yeah, that's actually a good parallel. That's also basically, in the sort of grand scheme of things, that's exactly the same paradigm, phase separation. Yeah.
But of course, actively translating ribosomes, they know where to go because they go for the RNA.
Well, they only go for the RNA when they get within a certain sort of physically attracting radius. But that radius is only a few angstroms. To find, that's just, I guess, enzyme physical chemistry. There's a sort of diffusion timescale where random collisions occur, and beyond that timescale, then you have the transition. Is that it? Thank you.
Okay. So we go on to Magnus Ivarsson. And he's going to talk about fossilized microorganisms from the emperor seamounts, implications for the search for subsurface biosphere on Earth and beyond.
Thank you. Thank you for inviting me to give this talk. I'm a PhD here at Department of Geology and Geochemistry at Stockholm University. And yeah, I will talk about fossilized microbes and some indications to search for them here, both on Earth and as well on other terrestrial planets, mostly on Mars, I guess is the most interest. And this is probably related to sample return and both where to go and what to do with the material that we find. So I will move around in both large-scale environments and small micro structures. How to
Push the space bar. Big bar.
Okay, so why are subsurface environments interesting? Very shortly, compared to other, I mean, there are a lot of ideas where we should look for life on Mars, polar ices, the soils, and so on. And subsurface environments have been primary targets for some time now, for some researchers, and mostly because of the protection from radiation. Also, the impacts and daily temperature changes can be destructive for organic molecules and life and so on. Another reason is that liquid water is probably it's not present on the surface. So if we want to look for liquid water, we should go underground. And if we look here on Earth, we know that subsurface environments can support microbes to great depths. I think we found microbes at several thousand meters, both in continental crust as well as in oceanic settings. One good example is Astrobio here in Sweden, the continental crust. And the only requirement seems to be water and the supply of nutrients. As long as we find water, they appear to be there. So, if we go to Mars, where should we look? I mean, the subsurface environment is huge. It's just the whole planet. So we have to narrow it down. We know that water is crucial for it. It's a requirement, and the only possible way to find liquid water that can maintain for a very long time is at hydrothermal settings, hydrothermal environments. And where do we find those? Well, they need a heat source. So the most, the only heat source is volcanoes, both here on Earth and probably on other planets. So hydrothermal environments are both interesting in two aspects, mostly because of their capability to support life in extreme environments. But also, and that is what I will talk about mostly, is their capability to preserve paleobiological information. By that I mean the high rates of mineralization favor the preservation of microbes and biomarkers and so on. So there are extremely good environments to search for fossils and traces for life. And as I said, we need a heat source to heat the water and drive these hydrothermal cells. And the heat source is volcanism. And on all terrestrial planets except for Earth, the heat source and the volcanism is dominated by hotspots. Mostly because all these planets lack plate tectonics. Here on Earth we find the volcanoes at plate margins, spreading centers, and subduction zones, and so on. But on Mars and the other, it's hotspots that are the dominating volcanism type. And we have some hotspots here on Earth as well. So they are of course extremely interesting as analog environments for volcanism on Mars and other processes that is associated with volcanism. Another interesting thing with hotspots here on Earth is that they are considered to be too limited, both in circulation depth, circulation volume, temperatures, and so on, to be able to support a subsurface biosphere. One reason for that is that hydrothermal events lack these macrophones that we found at plate margins. Bacterial mats are found, but no larger organisms like tube worms and so on. So they have been questioned to whether or not we can support.
I have a question there.
Yeah.
Wherever you dig deep in the crust on the Earth, it gets hotter when you dig. So there is no welcome needed, really.
Not on Earth maybe, but on
Probably
Mars.
on Mars as well, because there is an internal heat source on Mars, the same as on Earth in the decay of radioactive matter. So, in my view, there is no need for transportation of hot material. The temperature is there if you dig deep enough.
Yeah, you have to dig very deep in Mars, I guess, to get those temperatures. I don't. But if we look at it this way, in the question of sample return, we can't probably dig as deep as that. So we are limited to quite superficial environments. And for that we need some sort of heat source to heat up the water. I don't know exactly what depth water can be liquid in Mars, but my guess is that it's quite deep. Okay, anyway. Yeah, we have studied the largest hotspot on Earth. It's the Emperor Seamounts. It's the same, this is the Emperor Seamounts, and it's the same hotspot track as Hawaiian Islands belongs to. So this is the Emperor Seamounts, and from the bench, Hawaiian Islands continues. And we have the samples from Detroit, Intoko, and Koko Seamounts. And they are drilled basalt samples. We have focused on very veined and fractured samples because we think those are the samples where we can find the biostructures that we search for. This is an example. This is a thin section of a basalt sample. So previously these fractures have been circulated by fluids, hydrothermal fluids, and but since these systems of fossilized hydrothermal systems, so there's no circulation left. All the cracks and the fractures have been filled with secondary mineralizations. So the interesting area where we looked at is the transition between the veins, the fractured zones, and the veins in itself, the vein-filling minerals. So that's where the microbes are supposed to be found. And we found quite a lot of interesting structures. First of all, we found tunnel structures or galleries. These are quite known structures today, and are reliable textural biomarkers. They are found in all oceanic settings today, both in modern as well as in ancient, as far back as 3.5 billion years old ophiolites they have been found. So this is a vein. So the idea is that while this vein was circulated by fluids, microbes could be transported here and attached to the surface, and then they etched themselves in like this, producing these long structures.
So how big is that?
These are about between 1 to 5 microns
In thickness, yeah? And the
in thickness.
penetration
They can be
depth?
as long as 200 microns.
No,
So
microns.
some are really, really long. And opposite to these, which are only etched remains, we find these kind of structures, which are attached to the vein walls, and then they are preserved within the carbonates in the veins. And these filaments are found in a lot of different morphological types. You have this tube structure. We found branched structures, segmented structures, and the previous that have this bean shape, we call it. It's a small, thin filament with a large knob on its end. And these are found in quite abundance. And the most striking with these are that they have rim or iron oxides around them. They are very highly enriched in iron. And then, so this is the filament itself, and then they have a rim of iron oxides like this. Now I will come back to that. Another microstructures that we found are these cell-like globules. And these are found attached to zeolites, which are secondary minerals. So here, where we find these, the alteration of the rock has proceeded more than the volcanic mass. And these are also found together with filament structures. So the problem, as usual with fossils and fossilized bacteria, is to prove their biogenicity. And we have used a couple of methods to prove this. First of all, we have used SEM or EDS and done element analysis on it. And we have found that they are highly enriched in carbon. We have sometimes more than 50% carbon. And even more interesting is that this carbon is not associated with the carbonates. So this is element mapping. This is carbon and this is calcium. And this clearly shows that, for instance, this is a filament in calcite. So the carbon is concentrated to the filament, and the calcium is concentrated to the carbonates. So it's not carbon connected to the surrounding minerals, and this indicates that it could be organic. Another way we have used, another method is pigments. We'll stain them with pigments. We've used PI and Doppler staining, and then looked at them in fluorescent light. And PI was quite successful. We succeeded in binding PI to some of these structures. This is a filament previous staining, and this is afterwards. This is globules before and after staining by PI. And PI is known to bind to damaged bacterial cells, so this is a good indicator. Another method we've used is the PROSIMS, which I guess Sandra will talk more about tomorrow. And these results are a bit preliminary, but we found that phosphates, I can explain this. This is a filament, and this is a video picture of this filament. So you can see that phosphates, hydrocarbons, are concentrated to the surface of this filaments, and we also have some indications of lipids being preserved in these structures. But this is a method that needs more.
Where are those on the picture? Is it the dot in the middle?
Yeah, this is this official part. So this is taken in transmitted light, so you can see the whole filament at different cycle levels. But this is only the superficial part. So this is what we see. And this is also a cut through, a cross-cut through the filament. So this is a very small piece of it, but it's the only superficial part. Another method that we've used to maybe not prove the biogenicity, but to find some sort of characteristic appearance, we have used iron isotopes, and the method is laser ablation, ICP-MS. So we have obtained profiles through these filaments, cross-cutting profiles, and we have compared carbonaceous filaments, which are microfossils, structures we think are truly fossilized microbes, and we have compared them to non-carbonaceous filaments, which are probably abiotically formed filaments, but they are very similar in appearance. So what we get is this very characteristic profile pattern. I can explain it. The black is iron 56, the red one is iron 54, the green is iron 58, and the gray is iron 57. So when we enter the filament here, the outer rim, which I showed earlier, that contains iron oxides, iron 56 goes up like this, and the other isotope goes down. While we move through this filament and out of this outer rim and move more into the filament, iron 56 drops and the other goes up like this. And when we hit the next rim of iron oxides, these isotopes go down and iron 56 goes up. And this is the pattern we get from all microfossils. And when we compare it to these abiotically formed filaments, we only show a peak of iron 56 and small changes on the other isotopes. But they don't show any internal ordering like this. So this is quite exciting, but it's too early to use it as a reliable source to prove the biogenicity, but it is at least a very characteristic trend so far. And we need to look at other samples from other settings as well.
So the x-axis is actually the distance across the sample?
Yeah, this is the distance. So this is actually given in seconds. So this is the profile yeah, if this is the filament, we kept it through like this.
And what is the 13 to 12C ratio?
Sorry?
The carbonaceous isotopic ratio, what is that?
Oh, we were not able to do carbonate isotopes because it's unfortunately it's we can't look at the light compounds in laser ablation. We need a isolated well, box or something, but we don't have that yet. So it's only metals and so forth. So, yeah, so we are pretty sure about the bioenergy, at least. And what can we say about this, and why is this interesting in an astrobiological perspective? I think the most interesting with this is that we find microfossils in a big variety of niches, micro environments, which are produced by the alteration of the rock. For instance, these tunnel structures are found in volcanic glass, which is the fresh, unaltered volcanic production. And when we move up in the alteration to lower medium alteration, we find filaments attached to altered basalt and altered glass. And even further, in the high alteration zones, which are characterized by zeolites, we find these globules, as I showed you, as well as filaments attached. So this shows that as the alteration of the rock proceeds, the microbes can, well, I won't say adapt because they are fossils, but it's obvious that they can inhabit all these kind of microenvironments that are produced by the alteration. And if we return to Mars, we know that the volcanoes are, as I said, fixed at the same spot all the time. They are fed by the same plume of magma, and this results in extremely large volcanoes. Olympus Mons is the biggest volcano in our solar system, and if you compare it to our area of investigation, we can see that the whole chain that we have looked at fits within the crater. Another thing is that these volcanoes are extended in time. So Olympus Mons, for example, have been active for 80% of the life, of the whole lifespan of Mars. So they are active for extremely long periods of time. And it's, I mean, it's the question is, can these isolated systems support life, microbial life, during such long period of times? And if we look at our results, it's obvious that microbes can adapt to all the changes and all the alteration process that takes place as volcanoes evolve and are active and so on. So I think that's a very interesting result from this. So finally, I just want to conclude that hydrothermal systems associated with hotspot volcanism are capable of supporting a subsurface biosphere. And this is of course interesting both here on Earth but as well on Mars. And my opinion is that these environments should be very interesting to search for fossil records on Mars. That was what I had to say. Thank you very much.
Questions? Anthony?
Yeah, so just regarding the second conclusion of yours, do you think would that be your best pick of place to search over other? I mean, people have suggested subsurface and so on, but do you think that would be the best place to look for?
Yeah, I think so. Absolutely. I mean, that's where we should find liquid water during long periods of time.
Because of the sustained water.
Yeah.
How quickly would the water disappear?
You mean if the volcanism decreased?
No, I mean when. I don't know. I mean, okay. If you had an eruption, I guess it'd either seep back in, or something on the way.
That's hard to say. I mean, if it's in the early times of Mars, when we could have had water, an ocean maybe, so the water probably did never go away. But if we only have a subsurface, have a thermal environment, I don't see any reason. I mean, there has to be a source of new fresh water, of course, but that could be the magma could bring some new water as well. It's hard to say, actually.
Amen.
If you compare your fossils with these inclusions in the Martian rock, or what was the when you compare that, how similar are they or dissimilar? I thought there was a huge range or a difference in size, right?
You mean those found in this ALH?
Yes. The famous pictures of microscopes.
Well, they are filamentous as well, of course. I can't remember the actual size of them, but
They were 10 nanometers.
nanometers.
Yeah,
Okay.
Yeah.
These are
That was one of the arguments, actually. Yeah.
these are those are very small. These are around the filaments I showed you are about 1 to 5 microns thick and can be 50 microns long sometimes. They can be even longer, actually.
But do they show this pattern? I mean, it's a fascinating thing that these morphologically on these pictures that they really look like worms, because they have these they're very different things attached to each other.
Yeah, they were segmented.
Yes.
Yeah, we see similar things, but my personal opinion about those in that rock, I think it looks like cracks afterwards. I don't think they are a preserved structure, but that's my personal opinion. Another thing with those are that they are, I mean, they're found on the surface. These we see these in a thin section, so they are not superficial parts. They are within the rock still. The Martian meteorites were superficial. I mean, they were on the surface, so they can have attached to the surface any time, actually.
Okay, excellent. And Kai?
These structures that you showed in the very beginning, with a length of 200 micrometers, these are really traces of propagation. Is that true? Are these?
That's the interpretation.
So what is the speed of? So that propagate this, but it propagated prior to the formation of the fossil, or how? Or was it?
Yeah, it's
For fossilizing.
It's not a true microbial fossil. It's only the track.
Yeah, right.
So
Okay.
the idea is that they have attached to the surface and then protruded more and more and more. I don't know how long. They have done some experiments actually on volcanic glass, where they have produced similar structures, and they migrated 10 microns during a period of 6 months, I think.
So it can actually be seen.
Yeah.
Mm-hmm.
What migrated? What was the test?
They had volcanic glass and then microbes.
What kind of microbes?
I can't remember.
But
But they saw that they attached to it and etched the surface. And migrated.
Is that a chemical process?
Yeah, it's some sort of dissolution of the silicon aluminum framework of the glass. Some sort of they lower the pH below pH 3, which increases the dissolution. I don't know exactly how it works.
Right?
I think the intriguing part is that you actually find these isotopic patterns. I think fossils as morphological structures will always be debated, especially with there have been reasons, but also on in terrestrial samples it has been argued that cyanobacteria have been present 3.5 billion years, just on morphological structures which look like cyanobacteria, but then it was actually shown that these structures could be produced just chemically. So that all these old fossils are quite, they are questioned, but when you have these isotopic patterns that you see with the metals, that's I think this would be to go for if you have find isotopic anomalies that you cannot explain chemically. I don't know if this would be possible chemically, that you have this nice separation of the different iron isotopes as you've shown it. And if you could find this in a mass sample, it would be quite a strong, not evidence, but strong argument at least, that this could be produced by organisms. And actually you could test this in modern material just by the experiments that you have talked about in I think in the Bergen group of Theo Torsvik. They work with these kinds of organisms in volcanic glass, and they if you took some of the modern material and you could find the same, where you actually know that microbes have been etching and they may be still present in the cavities, and you could also find this iron isotopic pattern. It could even be a stronger support for your argument that it is biological and not chemical. And this could then also be used on material that is bone masked. I don't know if this Alan Hill meteorites actually have been investigated. But on this kind of, there have been all kinds of investigations, but I never came across investigations looking for iron isotopes.
No, I don't think so.
But there's no glass, right? That's not.
No, it's
There's no glass from Mars, right?
No.
But so could I ask you about the isotopic enrichment? And this is out of my ignorance, actually. Does one know the mechanism behind it?
No.
Because for other kind of isotopic enrichment, it's, I mean, chemically, a lighter isotope would be easier to metabolize than a heavier one. So then it's easy to understand. But I think some of your iron isotopes were actually lighter than the natural abundance. This
actually shows the opposite to what is expected somehow. I don't know the process behind this. The only thing that you can say is that the iron isotopes, that if those organisms are iron oxidizers and have precipitated these iron oxides as a process in their metabolism, it's possible to say that those iron oxides are enriched in iron 56 and that they have used it.
Mm-hmm.
But why the filaments themselves are enriched in the heavier...
But you had both 55 and 57, hadn't you, in the filaments?
54 is sometimes following, not following, it's not there. Actually. It's mostly 57, 58.
Okay, then it makes sense.
Yeah.
So I think it would be a strong support if you could show it in modern, or just in experiments where you see the same, where you actually have organisms actively etching this material.
Yeah, it probably needs more work.
And I think if they are going to actually have these kinds, to do these kinds of experiments, they would just ask them to get some of their modern material and use your method.
Yeah, I know that they are working with a lot of these tunnel structures in glass. So I have never looked at the iron isotopes in the tunnel structures, only in the filaments. So that could be another thing to do as well.
Okay, thank you very much. And then to our last speaker for today, Christian Lindgren, Self-Replicating Patterns and Information Dynamics.
Self-focusing, normally.
You can focus on
Yeah, just to do a preservation maybe. Yes. Oh, that's it.
Okay, thanks for inviting me, Axel, to this biology meeting. My topic is slightly off the central theme here, but I think there are connections to astrobiology. What I will present here is actually part of a larger European project that we are partners in, the project PACE, or Programmable Artificial Cell Evolution, in which we study the possibility to create lifelike properties in artificial chemical systems, properties like self-assembly, self-repair, self-replication, and evolution. And the long-term vision and sort of the driving vision in the PACE project is to create artificial cells that, from scratch, from bottom up, actually not being based on the chemistry that we know from biological systems, but something completely artificial. In the PACE project, we are taking the first steps in that direction. We are not going to finish that goal within the project, which actually ends in a year from now. It has gone on for almost 3 years. But there has been some steps along that route. I'm a theoretician, so I should mention that what I will present is theoretical studies on artificial chemical systems, but exhibiting some properties of lifelike behavior. I will also present, and that is this part of it, an older piece of work of mine that we have applied to chemical self-organization, actually developing an information theoretic framework that we want to use for characterizing pattern formation in chemical systems, trying to actually connect the information in patterns being formed with the thermodynamic constraints that the chemical system has to obey. And also, I also would like to advertise that in the PACE project, we did start a center in Venice, the European Center for Living Technology, 2 years ago. And this is now a center that is continuing to live on its own. It has received new funding. It will start up experimental activities also. So if you are looking for a place in which to hold a workshop in astrobiology, I like to suggest that you look for a possibility to do that in Venice. It's a nice location, and you can contact me. I'm currently one of the co-directors for the center.
Where is it situated?
It's close to the Academia Bridge, not far from some other place. So I will start with an experiment and a simulation that was presented 15 years ago, in the beginning of the 90s, where researchers at Los Alamos and in Texas showed self-replication of patterns in reaction diffusion dynamics. And that has inspired us to study that type of dynamics, both by extending the model to look for the possibility to use that in the PACE project, but also trying to use information theory and thermodynamics to get a deeper understanding on what is happening in this type of process. So I will then present what we developed in the 80s actually, but that we continue to work on now, which we call geometric information theory, that connects with thermodynamics. And that will be summarized in the continuity equation for information in chemical self-organizing systems or in pattern-forming systems. I will only give sort of an overview of that. And then I will discuss how we have modified the theoretical model for this self-replicating pattern into, modifying the mechanism and how that can be applied to a different type of reactor, a reactor that has been developed technically in the PACE project, which is sort of a lab on chip. Could we get this type of self-replicating patterns also in that type of confinement? So that will be the end part of the story. But let me start with the self-replicating patterns experiment that was done in Harry Sweeney's lab in Texas in the beginning of the 90s. He used an iolate ferrocyanide sulfite reaction to show that you could create concentration peaks in a simple system, concentration peaks that actually could divide into two new peaks, and that process could go on for a considerable amount of time. And the nice thing with those experiments, I think, is that they demonstrated that it's possible to have self-replication of macroscopic objects without having the usual confinement of a membrane or some other equilibrium type of structure that we have in biological systems. So I think this is a good example of that self-replication as one of the most important properties of living systems could at least happen in completely different types of settings than we are familiar with from biology. Just briefly saying a little bit about this, the experiment, it's done in a sort of a two-dimensional gel. It's a couple of square centimeters, a very thin gel, and it is in contact with a stirred tank reactor in which sort of the fuel is diffusing into the system from perpendicular perpendicular to what we see here. And the way I describe this in detail is that this is a pretty complicated type of experimental setup if you would like to perform experiments of this type in a lab on the chip, where it's difficult to arrange this type of three-dimensional setting. So in the PACE project, one has developed another technology in which you cannot really feed the system from top down, but you actually have to have a single point of inflow of the chemical components. So that's why we actually would like to look at other possibilities to create similar types of self-replication, but in other confinements. So I will get back to that to the end of the talk. The simulation of this type of phenomenon was also presented in the beginning of the 90s by Pearson, but also in the paper by Swinney and Lee. And this is a very simplified reaction scheme for the process that they did experiment on. But the active part here is really this self-replication or self-catalytic reaction where we produce one sort of abstract B molecule from a U molecule using two other B molecules as catalysts. And the system or the reactions take part in a two-dimensional square in the simulation. We have an inflow of the fuel molecule from top, and there is an outflow of B molecules downwards also. And then there is a decay process where B also disappears into G, and G is inert here, so it doesn't take part in the reactions. And we model this by usual reaction diffusion dynamics, and the nice type of behavior that we see is that if we start with the concentration peak of B molecules in the background of very low concentration of B, we see that this concentration peak stretches out in space, divides, and this process goes on. All the time here we have the U molecules flowing down, feeding the whole process, making all this possible, of course. You also see that there is some instability in the system. Some of the concentration peaks start to oscillate. When they die, they leave space open for other B molecules to continue their splitting process. So we get in this type of system the population dynamics on the level of concentration peaks. So these peaks, they split, they die, and new peaks can be born. So it's really a system that exhibits some type of lifelike behavior without having almost any similarities with biological systems.
Why they are not just getting bigger? Why do they get this elongated bean shape?
Why they get that? I mean, that is a pretty complicated dynamic feature. But I mean, this is, I mean, on the level of, I mean, now in this abstract model, on the level of molecules, this is the driving process, and then there is nothing else that sort of controls it. So the goal now for me is to present first our information theoretic approach to analyze this type of pattern formation process and how to connect that information analysis with the thermodynamic constraints of this type of system. But before doing that, I'd like to just mention with a few words the concept of information and how we choose to, or what type of information quantity we use to describe the pattern. Because there are many different ways you could approach information in chemical patterns. One approach that is strongly related to biology would be to talk about the information in the dynamics. I mean, information about what are the molecules involved in this process, what are the reactions involved, etc. And this would relate to sort of genetic information in biological systems, since the genetic codes for the proteins that tells what are the components that are active in the system. But this is not what we are going to focus on in the framework. Another important information component is information in small fluctuations in the system. Fluctuations that are necessary in self-organizing systems to cause a symmetry break, to specify exactly what type of pattern are we going to see. I mean, if you run the same simulation or if you do the same experiment in a specific chemical system many times, you will see similar types of patterns all the time, but they will be different from one time to another. And that difference comes typically from random fluctuations or small differences in the initial states in the different runs you have done. So that is information in fluctuations or noise. and we are not going to study that in this framework. It can be studied in connection with this framework, but we are not using it. But what we are looking at here is the information in the thermodynamic driving forces, the information capacity in free energy that drives the pattern formation process. And we typically have these relations that the information capacity in the free energy is much, much larger than the information in the noise or the information in the genetics of the system. But I wanted to stress this because otherwise when I present this, there can be a lot of confusion what type of information I'm really referring to. And if you want to use information theory to describe information in a chemical pattern, one of the basic concepts of information theory that works well is relative information, where you start with an a priori description of the system, and you then try to quantify how much extra information do we gain when we learn the exact, or when we observe the exact concentrations of the system. So I do this very briefly, but so if Ci0 are equilibrium concentrations of the components involved, that would be our sort of best guess if we don't know anything about the system, but then we make an observation, we see how the real concentration profiles look like for the different chemical components I. And then this sum is the information theoretic relative information quantity that tells us how much information do we gain by observing the actual concentration profile. And we integrate this over the space. So this is the sort of the direct application of information theory to a chemical pattern.
So this is just an entropy question.
Yeah, that's right. So if we just look at this term, that is the entropy term. So this is the entropy term we have in statistical physics or thermodynamics. When you have the a priori distribution in the denominator in the logarithm, you get the relative information or the relative entropy. But this, together with entropy, this is one of the basic concepts in information theory, which also is what you use in communication theory. So this is the total information of the chemical pattern, the chemical system, at a specific point in time. Now, we want to study this in the context of thermodynamics. And if we go back to our self-organizing system, I mean, in order for this system to be capable to build up some special structure and to maintain that structure, it needs to be an open system. So we have an inflow of fuel, light, food, free energy, and we need to export low-value molecules, waste, and heat, of course. And by having this system open in this way, we can, of course, maintain or actually even drive the system away from equilibrium towards a low entropy state and keep it there by exporting more entropy than we reduce in the system during the phase of build-up of the structure. So the second law of thermodynamics, of course, still holds, since in total, summing the entropy in the environment and in the system, we have an increase of entropy. But what we want to do in the information theoretic description is to view the inflow of free energy as an inflow of information capacity. And we would like to be able to see how this inflow of information actually gets distributed at different positions in the system and at different length scales to build up and form the pattern that we see. And at some point, of course, this information in the pattern gets destroyed by the processes of chemical reactions and diffusion, and information then leaves the system as heat or maybe as, primarily as heat, actually, I would say. So that is the goal. And the connection between free energy that is driving the chemical pattern formation process and information comes from this relation that holds in the case when we have ideal solutions in the chemical system. And we see here in this expression here we have the total information that I introduced from from information theory, and if we multiply that by Boltzmann's constant times temperature, we get the free energy of the chemical pattern. So there is this very direct relation between the information in a chemical pattern and the free energy stored in the pattern. So we have this relation that energy relates to information with a factor of log 2 and Boltzmann's constant time temperature. So it means that actually 1 joule of energy is in the order of 10 to the 21 bits of information. So if you, to put that in ordinary terms, it's like 1 piece of sugar contains an information capacity more than humankind has written over all history. So there's a lot of information capacity in energy. Now,
Sorry, what happened to the time?
Yeah.
I want to see the time change of the pattern.
Yeah, that I will get to that. But I now start with sort of the more of the frozen picture. But time will appear in a few minutes. The goal now with geometric information theory is to take this total information K of the chemical system and to divide that into contributions from different positions in the system and from different length scales. And then we would like to bring in time also to see how information comes into the system in the free energy, how it's distributed, accumulates maybe at certain positions when the spatial structure is being built up, and how this information then leads to system as entropy production at the end. I would like to be able to describe this flow of information in time through a continuity equation. But in order to be able to locate information at different positions and at different length scales, we need to introduce resolution, a parameter which we can vary in order to observe the system using different sharp glasses, or we observe it at different length scales, so to say. And we do this by introducing a resolution-dependent concentration description, where the parameter R is now a length scale parameter. So when R is 0, you observe it with length scale 0. That means you observe the exact concentration distribution. But when R is increasing, you sort of blur the picture more and more. So here is an example of the system observed at bad resolution, high value of resolution parameter R. And when we decrease R, we get more and more of the structure of the underlying picture, and by doing that we sort of gradually get more and more information from the system that we study. And so we do place our system, still no time involved, still just one frozen moment in time. So we place the system in space spanned by both position and the resolution parameter. And then we decompose the total information in the chemical pattern. We want that to be decomposed into contributions from both different length scales and different positions. And length scales, intuitively you can think that when we improve resolution, there might be some length scales, values of R, when there is a lot of new information being revealed. So if we look at sort of the resolution-dependent information, there might be some position here where we really learn the underlying structure, when we really see what the underlying structure is. And then after that we don't see so much new details of the pattern. And then one can also, I will not have time to go into the details of that, one can also decompose this resolution-dependent information into contributions from different positions. Just one very brief comment here is that this information density, which is then the final information contribution from different length scales are and positions x, this information density depends on the gradient of the concentration profile. So it means that information density is high when we have large changes in space of concentration, which makes sense if you think about pictures, that you would tend to say that information is at the edges of objects in a picture, and not when you have the same intensity, you don't say that information is there, but information is really located when it goes from black to white, for example, in a black and white picture. So this decomposition is also actually useful for picture analysis. Let me also say that the first decomposition I made here of the total information is done so that there is actually some information that we can see already when we have arbitrarily bad resolution, because even if we are not capable of seeing where in the system single molecules are located, we can still say that the average concentration in this system is different from equilibrium concentrations, and that would actually tell us that this is a system far from equilibrium. So even at the worst resolution, we can detect that there is some chemical information. This is a system that deviates from chemical equilibrium. And then the rest of the information in the system comes from different types of spatial variations. And that spatial variations is then decomposed into contributions from different positions and different length scales. And so this is now again the simulation that we saw before of self-replicating spots. In this picture, I show the information density. That is the contribution to the information from different position and at a certain resolution length scale, R being 0.01, which is a very fine resolution. So it means that at this level of resolution, we can detect single concentration peaks. So we see that the concentration peaks are reflected then by circles of higher information density, because it is at the edges of the concentration peaks that we get most of the information. But when we worsen the resolution, increase R, we see that the information density is not capable any longer to detect single concentration peaks, but there is still some structure that this information density can detect. And what it actually detects is that in some points in time, there are large areas that die out, so we get holes in the population of concentration peaks, and those holes are now seen at these larger length scales. So this information density quantity can detect patterns at different length scales. So here we start to see some time dependence in it, but I will only be able to briefly summarize the time-dependent picture of this information dynamics. So again, we span the space of position and resolution, and what I placed here is the information density of the system at various length scales, and the system is driven by an inflow of the fuel, and the inflow of the fuel is keeping the system out of chemical equilibrium. So the inflow of the fuel keeps up this part of the information in the system. Now, that chemical disequilibrium, that chemical information, is then flowing down in length scales, being accumulated at concentration peaks when they divide, but all the time flow is going down, and at the very finest levels of resolution, information is destroyed because of entropy production due to chemical reactions and due to diffusion. And what we have derived is a continuity equation that sort of summarizes all these flows, and I will not go into the details of that, but I can refer to papers on details if you want. I skip that, and I I now go on to describe how we have tried to modify the mechanism in this self-replicating spots system in order for it to be capable also to have self-replicating patterns in a certain flow reactor that has been technically developed in the PACE project. It's called the fan reactor, primarily because it looks like a fan. There's an inflow at one edge and there's an outflow at, inflow at one point more or less, and an outflow at the other edge. I mean, the important difference here is that we are in this type of reaction chamber, we are not capable of driving the system by an inflow of fuel perpendicular to how the system is stretched in space. But all the fuel must enter at this point, and that makes pattern formation more complicated, because the risk is that all the fuel will be consumed at the inlet, and nothing will be left for keeping structures maintaining spatial structures in the reactor. And this is one example of how this has been technically built. There has been some experiments done in the reactor, but not experiments looking at spatial structures in 2 dimensions, but only looking at standing waves, where one has used the distance from the inlet as the only variable. Now, what we have done in order to make it possible for self-replicating patterns to be also produced in this type of reactor is that we realized that we need to have a precursor to the fuel that does not directly become available for the reaction. So this is an equilibrium reaction between the precursor of the fuel and the reaction that is shifted far to the left. So it means that if the precursor to the fuel enters, it is kept back. I mean, there is a barrier for this precursor to be transformed to B molecules and to take part in the reactions. And this type of barrier is probably, this type of mechanism is probably important in many types of systems. I mean, we don't want fuel to be used immediately when it is there, but there must be some way in which how much of the fuel is used can be controlled or can be held back. So this is now a simulation example. Of that system. So we have the inflow of the precursor to the left. We see sort of standing waves in the direction of the flow reactor, but pretty soon we see that these standing waves break up into smaller concentration ridges or even concentration peaks, and pretty soon we actually see that we have concentration peaks splitting also in this type of confinement. Now, in the continuation of this, this is now done only in a very abstract model, so we are now discussing with John McCaskill, who is a chemist with the lab, and he is the one who developed this reactor. We are looking for possible real chemical components that could play this role to see if we could create this type of system also in the real fan reactor on the lab of the ship, on the chip. So, what I wanted to summarize for you today and describe is first the type of information theoretic framework that we have developed for describing and analyzing pattern formation in reaction diffusion dynamics, and then also very briefly this work that is not at all finished, not even published anywhere, the extended gray spot model, where we can study self-replicating of spots also in a flow reactor architecture, which is something that has not been tried experimentally before. Thank you for your attention.
Thank you very much. Axel?
So is the main advantage of the flow reactor actually that the reaction happens, or the flow, or the fluxes of energy actually happen in the plane, as opposed to perpendicular to the plane? In the other models you had the energy input and losses perpendicular.
Yes, that's right. So that's more tricky to
so this is more realistic
to arrange,
yeah.
And also more realistic, I would say. I mean, you may have sources of energy located at different positions, and then that can be used by some non-equilibrium process. But also one advantage with this fan reactor, technically, is that across the, if you look at across this area, you get different characteristics. So as we actually saw in the simulation, we do get spots replication at a certain distance from the inlet. So in the same reactor, we can actually at one point in time study a large variety of chemical conditions. So that is also one advantage.
But isn't that dependent upon the time or the equilibrium of the precursor to the U?
Yes. Yeah,
because that in effect is how far your reactive molecule will, or your unreactive molecule will come before.
Yeah, that's right. So by varying that, probably by varying the concentration in which the precursor enters, you will vary the distance where things start to happen. But in many cases, I mean, there might be other things other concentrations you could vary also, where you don't really know what you should choose. And by running a single experiment, you may actually observe, okay, now we actually did get something happening on that distance, and you may learn something from that single experiment. You sort of capture a large number of chemical conditions. And there's also another thing that you have different flow velocities. I mean, there is a laminar flow here. And so the flow velocity is much faster at the inlet and slower over here. And that is also causing different conditions for pattern formation.
Nicholas?
Yeah, I wondered, because in equilibrium thermodynamics you do have phenomena, critical phenomena, where there's no particular hierarchy of length scales. I wondered if theoretically you have that prospect in reaction diffusion dynamics.
I would like to be able to do that, but in reaction diffusion dynamics, I mean, what you refer to are, I mean, would for example be lipid aggregation to membranes, etc. Where you, I mean, where the equilibrium processes cause some spatial structure.
Yeah, well, that's one aspect, but there's also you showed a very nice sort of projection of information content on the various length scales.
Yes.
And you have in your particular models, you have a well-defined peak at a certain molecular length scale. So I wondered if you can break down that hierarchy so that actually information is fairly evenly distributed across all length scales. Which, and those classes of phenomena do exist in equilibrium thermodynamics, close to any general phase transition.
Yeah. But I don't think you will see that in reaction diffusion dynamics.
Why is that theoretically impossible? Is that the issue?
Because of
maybe, I mean, it's a good question. I mean, if one would drive the system, I mean, appropriately. But yeah, I mean, I'm not going to rule it out.
Maybe it's because the nonlinearity is completely local. There's no gradients involved in the types of nonlinearity. If you had advection diffusion, maybe, then maybe it's different then. I'm just thinking of the analogy or lack of analogy with turbulence, of course. There's the nonlinearity of a different form. That's for U.God Utah type. There you have that, of course.
Yeah. But in principle, I mean, the simulation we saw on the self-replicating spots, we did have some information on the level of on the length scale of the spots, but then there is also some information when we get holes in the population of spots. So there's information also there. I mean, it could be that we have information also at higher hierarchical levels.
Well, is it an archetype? Is there a problem with your definition of information? If you were to sort of revise that definition, would you have a...
But what I mean, what you refer to as these, I mean, close to phase transitions, etc., would be if we look at, for example, spin models, like the Ising model.
Yeah.
In that situation, you would use a different, a very different, I would say, information quantity, because then you identify information with each state in a local cell.
Yeah.
And then you actually also, in that case, it's also very important to take into account correlations between different positions. Here, I'm not using correlations.
No.
Correlations are left out here.
Correlations are sort of arbitrary measure of information content. They work in a simplified world of equilibrium phenomenon. But in biology, you have obviously you have different length scales of organization.
Yes, but I mean this framework is very simple, and I say it only applies to reaction diffusion dynamics. So that's a severe limitation. Well, we are now extending it also to include advection, but that will not make a very big difference. So and the big disadvantage is that in this framework we are not really capable of treating equilibrium structures like micelles or vesicles in a good way. We don't really know how to do that because that would not be captured in reaction diffusion dynamics in a good way if you want to make an information picture. So it's, I mean, I would say there is a limitation on what this framework could be applied to. I think it can be applied to this type of chemical patterns on the macroscopic level, but when you get to microscopic or mesoscopic structures, I'm not so sure.
These are your patterns are mesoscopic.
Oh, yeah, okay. What is okay? But I almost would call them macroscopic. I mean, patterns are on the length scale of a millimeter, or close to the millimeter.
Okay,
length.
Rafael?
Yeah. Okay, lots of problem with the terminology you're using, and I don't really understand what is the point in calling the entropy information. Isn't it? I think it means much more confusion than anything else. So I barely see no information there. So why using these terms rather than entropy? We know what entropy is, but information we don't really know what it is.
So you're saying it's just an ordinary thermodynamic entropy?
Yeah, but
maybe it is actually then.
Yeah, I mean, but that is what I that's why
what you're actually trying to say,
because
right?
I mean this is I've heard this comment many times. So that's why
actually it's not true. It's not the same.
It's not the same. So that's why I actually start with this first three different information perspectives one could have, and I say that what the information perspective we have is to look at the information capacity in free energy. So that's the starting point. And of course, we could call this free energy, but what we do here is that we generalize, in that case, free energy to, and we decompose free energy into contributions from different positions in different length scales. Okay.
Yes, but why information?
The reason why we call it information is that there is an information interpretation of this. I mean, if you would apply this to pictures, for example, this would actually tell you how much you can compress a picture. Using, I mean, this has a direct connection with information theory, applied also to other fields of science.
Yeah. Okay, but maybe in the astrobiology context it may be confusing because we may think of this information in a relationship with the genetic information, and maybe this is quite different.
Yeah, that's why I put the first slide where I said this is not the genetic information.
Yes.
Because information, I mean, and this is one of the things I always start with when I teach information theory, that what the information theory concepts mean depends exactly on what probability distribution do you put into those quantities. So if you choose a certain distribution, it will be genetic information. If you put another description or of the system into it, it will be something that's related to free energy. But yeah.
I think it's a terminology problem. I think that when we listen to all these terms of information everywhere, when they just think that okay, but this is energy, free energy, or entropy, this is much more clear.
But the point is that it is not free energy, because since we decompose the total free energy into contributions from different length scales and positions. I mean, I could call it whatever I want. I mean, if I just make clear what it means, then I don't think it matters so much what to call it. The reason for calling it information is that it has a counterpart in information theory directly.
Are your numerical experiments always that nice? Or do you encounter runaway and freakouts, and you say you are far away from equilibrium, so one might naively expect that at times you get runaway effects. No?
I haven't tested
they
this.
Don't look like they don't look to me like fractals either. So I try to get the idea behind it. You don't see fractals.
No, we don't see fractals in this type of system. Actually, you may see fractal patterns in some self-replicating systems.
Well, they have a fractal index, but it's just not very impressive. It is a fractal structure, but it's got very low fractal.
But it would then be a fractal where the dimensionality depends on the resolution more strongly, I would guess.
As always, yes.
Yeah, yeah. Except for mathematical fractals, where it may be constant for resolution.
Yeah. I mean, the coastline depends.
Yeah.
Oh, you mean the dimensionality is the same? Yes.
Yeah.
I see. That's right.
Yeah, but there's always a nicely behaving. I mean, is it because you are tuning the
No, I think
boundary conditions, or
the nonlinearity is very nice to you.
Yeah, I mean,
It's stabilizes
well,
immediately always.
I mean, as soon as I have an underlying model for reaction diffusion dynamics, I can apply these quantities, and I mean, so the information picture will just reflect, or will just give you a different picture of what is happening, but in terms of information density or free energy density, if you want.
what I'm asking is related to my previous question during the talk, and that is I was trying to figure out what's the dissipation timescales in the system. Now, how fast do you pump the stuff through, and how fast do you actually distribute the information? This was actually my question. But
okay, I mean, the dynamics is fine-tuned in the sense that in order to get interesting self-replicating behavior of the patterns, you need to not drive the system too strong or not too weak. I mean, if you do that, I mean, if you drive it too strongly, I mean, it will run away. I mean, all the patterns will disappear, and we will have a very boring sort of burning the fuel to waste product directly. Or if it's too slowly pumped, I mean, it will go to a station or a state that is also very boring. So the how strongly you drive the system is a very important parameter that you need to fine-tune to get something that is interesting.
But isn't this then dependent on the rate constants for you wrote up in your operations?
Yes. So the rate constant.
You had a number of rate constant, I assume.
Yeah.
And so, in order to do something practical with this, I mean, you would have to know the detailed rate constants.
Yes.
In order to know whether it's going to follow this nice pattern or something.
Exactly, and one would. And also, I mean, this simple reaction u plus 2v goes to 3v doesn't hold. I mean, there's a more complicated reaction scheme behind that. And I don't even know if, I mean, the experiment that was done in the beginning of the 90s, I don't know if they have really figured out exactly what is happening. So this simple scheme is something they realize that it has the same phenomenology. So I think there is a lot of work to be done in order to understand the connection between that scheme and the real chemical reactions.
Okay, so thank you again. It says in the program now that we should have a half-an-hour discussion, so general discussion. So are there any questions that have popped up in your minds during today?
Well, I have another question on the last talk. So that
Okay.
seems to be one aspect of artificial life. Beyond that, what about issues like heredity? What are the prospects for carrying through the replication of the
evolutionary aspects of this? That's a good question. Now we are also looking at that. I mean, when we run this type of simulation with self-replicating spots, of course, you start to think about, I mean, what if the V molecules that are active within each spot, if they could mutate to something V prime, for example, if that happens within one spot, and that V prime sort of has a local selective advantage within the spot and take over that spot, then the question is, on the level of this concentration peak, the spot, will that concentration peak now self-replicate faster than the other peaks? And in that case, we actually will have selection on the level, not only locally in the spots, but also selection on the level of different concentration peaks. So we have investigated that, and you can actually see that mutations on the level of molecules lead to different selective advantages on the level of concentration peaks. Now, there is a problem in that type of evolutionary system, which is that you may get the mutation into a V molecule that actually destroys the concentration peak. And the problem is that this mutant may then easily spread. I mean, there is a risk that it may easily spread over the whole system and destroy everything. Mm-hmm. So this indicates that there might be a problem to resist parasites or resist mutants that actually destroy this macroscopic self-replication. And this problem has been studied before also. I mean, in the beginning of the 90s, Borlist and Hocheweg discussed resistance against parasites in spiral waves using the hypercycle. They used the cellular automaton model for that simulation, and then they could show that actually the spiral wave structure could resist parasites, because if there was a deleterious mutation within the spiral wave, that spiral wave would die, but the parasite would not be capable of entering other spirals. But I think, and it seems so, that to a large extent that result comes from the fact that they use the cellular automaton simulation method for it. As soon as you go to partial differential equations, it will be very difficult to keep parasites out of other spiral waves. So I would say that the resistance against parasites in this type of system is an unresolved question, and this may be the main disadvantage in a biological or in an artificial life setting, that the concentration peak as an entity doesn't have the barrier like a cell membrane, which may be a very big problem in an evolutionary context.
Mm-hmm. But parasites are real.
Yeah.
So what you have to imply is then you have to have, when once the system collapses, then you have to allow it to reappear de novo.
Yeah.
Somehow.
Yeah. Yeah.
So well, that's maybe different from real life. But
yeah. Yeah.
I mean, artificial life, it's possible.
Yeah.
You have parasites, they kill the population, they die off, and then in nature, then they may be eliminated from this area, and then they migrate. New organisms migrate into the area, and then.
I agree. I mean, I think as soon as if there are some spatial separation, so that when a parasite wipe out first a single concentration peak and maybe a subpopulation, that there remain other areas that still survive, and in that case, it could still work.
And because you have uniformity of your inputs. So I mean that's also different from real life.
Yes, that's right. So one could have the non-uniform input.
Right.
Yeah.
This is probably a very simplistic question. It's really not my field, but do I understand it right? That is the fan reactor, is that a three-dimensional model? In spatial sense?
It's a real reactor, so it's three-dimensional, but it's very thin. So it's.
I wasn't really sure how it is, because you saw the fan, but you also showed the round thing.
Yeah, okay. That was the round thing was sort of schematic ideas that has been brought up to have maybe a reactor where you have inflow in the center and outflow at the edge. But the real implementation is a fan with 90-degree.
Relay stuff.
Yeah, and it's very flat. So you have laminar flow, and they have done both experiments and computer simulation to show that the flow is really laminar.
Has the been work done in 3 dimensions to try to simulate? Would it be? I imagine it would be a big difference.
Mm-hmm. Yeah,
did you spots in 3D?
Yeah, Claes Blomberg grew up here in Stockholm when he was a professor here, and he has a PhD student to work with him on such things. So they did study self-replicating spots in 3 dimensions, and I don't now remember exactly how, but you I think, as far as I understand, it's more. You need to fine-tune parameters the higher the dimensions. I've only worked in 1 and 2 dimensions. In the 1 dimension it's very easy, 2 it's a little bit more tricky, and I would guess 3 is much more tricky, but they did succeed in it.
That would be actually the realistic case, right?
Well, I mean, if you have,
does it really happen in essentially in 2D?
Yeah, it works like in 2D because the thinness of the reactor is much smaller than the size of the spots. But you're right that the reactions are still taking place in 3D, but I'm not sure if you really need to go to 3D simulations.
Because you said they were pretty big, right?
Yeah. The question is how well the 2D simulations captures a projection from 3D, and if the length scales of the structures that are active, if that length scale is much larger than the thinness of the reactor, I would suppose that it would work very well with 2D simulations. But I guess I would need to look at that more in detail too.
Okay, Anthony.
I was just thinking about this parasite thing in the Hoverwig work. So when you were saying that you thought your model would be vulnerable to parasites, you were meaning this one with the top-down dribbling. Were you meaning that as well as the fan? Because I'm wondering if the fan is a directionality.
Okay, the fan I haven't tried.
Because you think, you could dominate an entire wavefront, but you may sweep them away as the reaction is being pushed in one direction from the source. I don't know, but I just wondered.
Yeah, that's a good point. I don't know.
Okay.
I will look at that, because I have not given up sort of looking at evolution in this type of system.
I think, the very clear thing with the Hoverwig thing is that it's because they were spatially constrained, and the ability for them to grow is inherently constrained by using a cellular electronics. So one wonders whether there would be plenty of different types of solutions where if there's some sort of spatial constraint, so you don't have perfect mixing, then you will be able to create the environment would be conducive to survival in the face of parasites.
Yeah, I think you're right.
But in order to play this game, you would need different components in this, more components in the system.
Yes, that's right.
Actually, a number of components to have, which of course basically contain the information. Then, of course.
Yeah.
Which is one vulnerable to parasites.
There are like DNA amplification systems that one could possibly use here. When I discussed this with John McCaskill, he thinks that there might be possibilities. Well, maybe not to have a lot of evolution, but you may have several different types of what would correspond to the D molecules at least.
Yes.
Okay, so we try again. Anyone that has come up with a more general question? Axel?
Yeah, the one thing that we didn't really hear too much about, and I brought it up when Leonard was speaking, is the question of life detection in situ. And I was actually thinking maybe Kai could have a response on that. I mean, we want to build equipment, of course, that is eventually able to detect life in a way that is much more reliable than it was back in the old Viking days. So have we made much progress since then?
Yeah, well, I think there. Well, I know that people working with NASA, they develop systems that, just on the Phoenix, they're much more focused now, going from the more geological and mineralogical types of research that they did on the rovers. They go more for the biochemical, and they just the first thing is to show that there is organic material. After all, there's no
without looking at metabolism, but
no, it's just showing that there is any, because until now Mars is totally sterile. There's no evidence for organic material, at least not from the experiments that have been done until now. Well, there is methane, which is of course organic material in the atmosphere, but not from just from micrometeorites that should be sediment on the surface of Mars. There should be organic material present there. It's about this. There's quite an amount.
Totally there, and it's not there.
But it has not been. I think this is the first step to show that it's there, and then also come up with some kinetics, how quickly it is disappearing again. And as far as I know, that people working in collaboration with NASA said they developed equipment to show or to measure amino acids at very low concentrations, which is the first step. And I think with the Phoenix, it has some of the same capacity as Viking. It can dig about half a meter into the ground.
Are there differences depending on whether you can have something that's shifted away from equilibrium, which would indicate potential for present life, and then you have these more molecular fossils like the geologists would find would be argued to be far too complicated, far too complex to be produced pre-biologically long hydrocarbons and so on. So isn't it a problem that there is such a diversity of methods that you could potentially apply? I don't know if it's really one question, is it? I mean, if you're looking at extrasolar planets as one completely separate set of approaches, and then if you're digging in soil, you've got geological approaches, you've got maybe more biochemical approaches, and then perhaps more molecular biological approaches as well. I don't know if there's is there a good one way? I don't think there is there?
No, not really. I mean, different. You mentioned amino acids, and the one thing that hasn't been mentioned is detecting DNA directly, of course, which could also be, of course, from that, maybe in the permafrost or something.
But you're making a lot of assumptions if you're detecting DNA.
Oh yes, at least it's much more. It's very sensitive, of course, but it's very specialized. Yes.
But are there any groups in the Nordic countries involved directly in developing systems of this type?
I mean, what about Villerslev? Is he anywhere close to that?
Well, Esker and I, as you know, are both skeptical to that as a starting point for looking for life, because it's so easy to contaminate. I mean, the approaches to look for DNA work very well if the material is abundant, if there's lots of DNA. But if there's very little DNA, chance of contamination becomes very high because you're using effectively an amplification mechanism. So it's very easy to amplify something that was a contaminant that you put there rather than something that you're interested in studying. So people like Esther work, for example, on permafrost samples, looking for evidence of very old either microbes that are still living there or dead microbes, for example. And the single biggest problem is contamination, simply because microbes are everywhere. They can easily get into the sample, and if you're using an amplification mechanism, then you can easily amplify up the wrong thing.
I mean, even on Mars?
I think especially on Mars.
You mean, if you're saying microbes are everywhere?
It's not impossible.
You mean they brought them from Earth with a detector, yeah?
Result. You need a result that would say that for several different types of measurement, you're getting consistency. So, for example, if you're finding a correlation as you go down through from the surface to the subsurface, you see a correlation between, say, the abundance of DNA, you can measure the concentration, and then the diversity, so that it perhaps increases for a bit and then decreases as you go too far down. And then, for example, in principle, in the cells that you saw, the same correlation with a viable cell count, so that you're seeing several different parameters all giving you the same indication, then that might be a way of doing that. But it's quite a sophisticated experiment. I mean, it's much better to be doing the sort of things that Kai was talking about, looking for biochemicals that could be more easily analysed. I think the last thing you want to do is look for DNA at this stage. Not that you shouldn't look ever, but it wouldn't be the first choice.
Yeah. The other application except Mars is, of course, the upper atmosphere. I mean, the first 100 kilometers up into the air. That's also quite unclear, I thought.
You mean in Mars? Or?
No, on the Earth. I mean, what about what is the biological activity like in 40 kilometers height?
Well, there are examples of microbes that are, at least people pretend that they have isolated organisms that hide in the stratosphere.
And that's reliable, yeah? Or kind of, almost.
Yeah, well...
some of it isn't. Some of it may be.
So why do you want to explore our atmosphere?
I mean, because it's already rather controversial what happens in 40 kilometers height. And the other advantage is it's very easy to do, of course. You don't need to go to Mars. You can basically do it at any time of the year.
What do you mean that this would be something that was brought into the atmosphere?
That, of course, I don't know. I mean, that's one possibility, of course, but it could also be brought up from the bottom, of course.
Yes.
We need to understand that.
Yeah, but there is a controversial paper by Winger, I think.
Yeah. Victor Massinger, of course. That one is
yeah, but this one is really special. Because he argues that the fact that you find organisms at I don't know how many kilometers, but quite remote, is actually evidence for transgression.
Yeah. I mean, that's the problem, of course. I mean, if nobody else cares to double check this, then we have such unconfirmed claims out there, of course.
Was twofold. One was a lot of people didn't like the conclusion, but the other problem was that some of the people he was collaborating with didn't believe the results, and so they actually wrote a letter complaining about the quality of the paper, as well as other people arguing that the results of the interpretation was questionable. But it's another case of it's probably very low biomass, which means that any of the methods that we use for studying organisms you're really prone to contamination. That's the worst problem.
But we have to face it at some point and have to
work on that. There's ways you can do it for certain materials.
Yeah, it's so much easier, isn't it? That's low budget.
From S-range.
They're happy to send up.
Maybe it comes out from this conference. It's liable to collect the bugs up.
But that's what they did. That's what these Wickram things they sent. The plates are open bugs on plates.
Yes.
Or, you know, it's very uncontrolled.
But
And
it's
instead of
related.
like-
I think it is related with your experiments in a way, because now I was thinking, well, can you derive or determine or estimate, put it this way, a growth rate in your soil?
I think there's no growth.
If so, where?
I would guess well, it's
But the thing has to do with, we were before already talking about this this clump, this meteorite from Mars.
Yeah.
So it's feasible that also meteorites have fallen down from the Earth on Mars.
Yeah.
And these are likely contaminated, and there have been experiments which show that these bugs are surviving harsh conditions, even in interplanetary space. So then Mars should be contaminated, but there are not even frozen margaritas there. So it should flower, the whole planet should be.
Yeah, well, I think it's contaminated by the aircraft or the spacecraft that have already landed there. I think
That's true.
Yeah, it's impossible to sterilize things 100%.
Okay.
So even with the clean rooms, and there has been quite a lot of work. But the problem is always, when you want to show something which is there at very low quantities, and then you don't believe the result because you always fear contamination.
Mm-hmm.
That's the problem. When we expect very little biomass on Mars.
Yes.
And and then when we find biomass, then you always have to somehow be reluctant and say, well, this may be also contamination. If it's not totally different. But in your paper you even state something that is totally different could even be a earth contamination.
Well, if the sequence was unique, it doesn't rule out that it's not Earth-based contaminant, because what we know is that we've sampled a very small fraction of Earth biology. We don't even know what we're on the spacecraft. That's a fact. So given that we don't know which bugs are hitching lift on the spacecraft, because we know that all the clean rooms are contaminated, and all of the methods in place to decontaminate the spacecraft don't decontaminate them 100%, and that it's been demonstrated that certain bugs will survive the conditions for transfer for the space flight to Mars. Then that's obviously going to be.
But I think it's a question of standard experimental error. I mean, of course you can 100% detect against contamination. But when you do your error analysis, you have well-defined thresholds of error, and then you can look for the significance of your results.
Yes, yeah, it's not an insurmountable problem, but it's probably the case that you run into the difficulties of, with the material, it's sufficient to do a study that allows you to distinguish between contamination and a real result.
If the only problem is the very weak signals, right?
Yeah, well, the signal may be so weak that you can't. You have a situation where you don't have additional information to exclude a contamination, and that's the problem. That probably the biomass, if the biomass is so low, you're stuck with that situation. You don't have enough strength in your experiments to exclude the null hypothesis, or to reject the null hypothesis.
Couldn't you determine the DNA sequence and see if it fits with anything existing or not?
No, because we still know, and people like, this Craig Benter that I'm sure you've seen in his swimming trunks on the pages of Nature, go off and sequence seawater, and they find completely new genes. We haven't even scratched the surface of biodiversity on Earth. Finding sequences that don't have genetic code won't help us either, because we know there are sequences where you don't find the genetic code and they're biological sequences. So there's a lot of problems with saying, oh, it's a novel sequence, therefore it must be real and not a contaminant. Until we have enough of a sample, which then that wouldn't be enough information on its own.
Right-handed DNA would be fine.
Yeah, right-handed DNA.
Right?
Yeah.
Don't worry about contamination.
I mean, chirality is a really good potential biosignature.
That would be just too good to believe it. You won't believe it because it would be too good. Somebody put it there.
Okay, I think it sounds like we're getting closer to a reception discussion than a discussion. So thank all the speakers for today, actually.
Thanks, Philip, for doing the